can grow it under the sterile condition. I told each and every step we have to maintain the sterile condition. Then we can ultimately get a whole plant, right?
The plant part used in culture is called as explant. This property of plant cells to give rise to a whole new plant is called totipotency. The term totipotency was coined by Stewart in 1968. Totipotency. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators, such as auxins and cytokines.
Totipotency means from any part of plant can able to propagate as a whole plant. That is, each and every cell have the potential to produce as a whole plant, right?
Technique of tissue culture. The first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Tissue culture is performed using a small part of a plant to develop a large number of plants in a short duration. It is thus also called as micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called soma clones. Micropropagation is extensively used in horticulture and floriculture and is employed in the large-scale production of tomato, banana, apple, etc. This technique can also be used to propagate high-yielding disease-resistant varieties in a short span. Even in conditions when a plant is diseased, tissue culture is used for recovering healthy plants from diseased plants by taking the explant of the apical or axillary meristem. This is done because the meristem is always free from pathogens due to lack of vascular supply. The process of using the meristem for propagation is known as meristem culture. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. There are many advantages of tissue culture, which are as follows. A large number of plants can be grown in a short period of time. Healthy plants can be grown from diseased plants. Plants without seeds can be multiplied. It is useful where sexual reproduction is not possible.
Hope you understand the basic of plant tissue culture.
Tissue culture. First, let us see about the culture vessels. The culture vessels used for plant tissue studies includes Erlenmeyer flask, that is a conical flask, Petri plates and culture tubes, 25 into 150 millimeters. What do you know about culture medium? The important media used for all-purpose experiments are Murashige and Skog medium, that is MS medium, Gamborg medium, that is B5 medium, white medium, that is W medium, and niche medium. The culture medium is closed with a cotton plug or an aluminium foil sheet. The pH of the medium is adjusted to 5.8, that is the acidic range. How is the process of sterilization done?
Don't worry about that sterilization or culture medium preparation. I will explain you, right? What are the components we have to add in the culture media, mainly the MS media, and how we can sterilize the media and how we can sterilize the tools, glasswares, right? Each and everything I will explain you, right?
Tissue culture. First, let us see about the culture vessels. The culture vessels used for plant tissue studies includes Erlenmeyer flask, that is a conical flask, Petri plates and culture tubes, 25 into 150 millimeter medium, white medium, that is W medium, and niche medium. The culture medium is closed with a cotton plug or an aluminium foil sheet. The pH of the medium is adjusted to 5.8, that is the acidic range. How is the process of sterilization done? Sterilization is the technique employed to get rid of the microbes such as bacteria and fungi in the culture medium and
What is
plant
this?
tissue.
What is this? What is this equipment? What is this?
Incubator.
No.
Laminar outflow.
No, this is not the laminar flow. She is keeping the glasswares into this. No, not autoclave. Dry oven. Oven. The glasswares and the tools are sterilized using oven. Okay?
So, it is important to sterilize the culture medium and plant tissues. The culture medium can be
autoclave
sterilized.
actually, but there are different types of autoclaves are there, right? In the department, the laboratory also we have autoclave. In the tissue culture laboratory also autoclave is there, right? But different forms, right?
By keeping it in an autoclave and maintaining the temperature of 121 degrees Celsius for 15 minutes. The plant tissue, that is inoculum, is to be surface sterilized. What do you know about chemical sterilization? By treating the inoculum in any one of the chemical sterilisant such as sodium hypochlorite, calcium hypochlorite, mercury chloride,
Mercury
for
chloride
50
actually we can't use nowadays, right? It was banned since it is a heavy metal. But if we use it, that earlier we use it with a lot of precaution, right? We have to wear gloves while we handle that mercury chloride, but right? But it is good sterilant, right? For that. For the woody plant part, if you obtain as an explant, the mercury chloride is the best choice for sterilization, right?
to 20 minutes, followed by a repeated washing in sterile water up to 3 to 5 times. What is inoculation? The transfer of explant, that is, the root, stem, leaf, etc., onto a culture medium is called inoculation. The inoculation is carried out under aseptic conditions for which an apparatus called laminar airflow chamber
This
is
is
used.
laminar flow, right? This is a large laminar flow.
Flame
Here,
and cooled forceps
this is the she is working under the laminar flow, right?
are used for the transfer of plant materials to different culture media kept in glasswares. How is incubation done? The culture medium with the inoculum is incubated at 26 plus or minus 2 degrees Celsius with the light intensity at 2000 to 4000 lux, that is, unit of intensity of light, and allowing a photo period of 16 hours
This
of
is the
light
growth flow, right?
and 8 hours of darkness. Induction of callus. Due to the activity of auxins and cytokinins, the expand is induced to form callus. The callus is an unorganized mass of undifferentiated tissue.
This is callus,
The mechanism
right?
of callus formation is that auxin induces cell elongation and cytokinin induces cell division, as a result of which masses of cells are formed. What is morphogenesis? The formation of new organs from the callus under the influence of auxin and cytokinin is called morphogenesis. Roots and shoots are differentiated from the callus. Such embryos are called somatic embryos, which results in the formation of a young plantlet. Two types of morphogenesis. They are organogenesis and embryogenesis. Let us see organogenesis. The formation of new organs such as shoot and root is known as organogenesis. The development of shoot from the callus is called cortogenesis, and the formation of root is called rhizogenesis, respectively. What do you know about embryogenesis?
Under the organogenesis also, direct organogenesis is there, indirect organogenesis is there, right? But I'm not going to teach all the things. It's unnecessary to you.
The formation of embryos, that is, bipolar structure having a shoot and a root, from the callus is called embryogenesis. These embryos arise from somatic callus tissue and are called somatic embryos or embryoids or somaclonal embryos. What do you know about hardening? Exposing the plantlets to the natural environment in a stepwise manner is known as hardening. Finally, the plantlets are gradually transferred to the soil. Discuss the status of tissue culture technology.
Right?
Hello and welcome to the sterile tissue culture technique video. Before getting to work with tissue cultures, it is important.
So before entering into the laboratory, tissue culture laboratory, you have to wear the what? Laboratory coat.
To wear personal protective equipment such as a lab coat, gloves, and closed-toed shoes. Lab coats should not be removed from the tissue culture room and should be changed and washed every few weeks. Please check with your principal investigator for any additional personal protective equipment and review the material data safety sheets for any reagents that you are planning to use. Prior to putting on gloves, wash your hands to remove dead skin cells and kill any bacteria.
Before entering into the laboratory, right, there is a small room in front of that. right? We have to hang the laboratory coat in that, right? So we have to wear it and we have to wash our hand using any hand washing, right? Hand washing detergent, mainly the T-pol is used. Whatever the hand wash, we can use it to wash our hands.
Yeah.
If you want, we can wear the gloves as well.
Turn on the UV light for 10 minutes. Once the 10 minutes are up, turn on the blower.
This is laminar flow, right? If you want the UV lights, we can on it, right? And we can't see the UV light, right? Soon after we on the UV light, we have to come out from that place, right? Thereafter only we have come in and off it, right? If not, normal fluorescent light is enough. We have to on the normal fluorescent light under the laminar flow.
30 minutes. During this wait time, you can bring media or other reagents to room temperature if necessary and spray surrounding benches with 70% ethanol.
And the rest of the places where we are working, we have sterilized with 70% isopropyl alcohol, right? We have to wipe it.
After you have left the blower on for 30 minutes, you can start working. Before doing anything in the biosafety cabinet, wipe down all surfaces with 70%.
What they are spraying is actually alcohol, 70% alcohol, right?
Ethanol. Make sure to spray down the working area thoroughly. Additionally, spray all items going into the BSC with 70% ethanol. Do all work 4 inches into the BSC and avoid leaving anything on the grill. Make sure you spray down your gloves every time before your hands enter the BSC, especially after touching something that may have been contaminated. When working with well plates or stalks of tissue culture dishes, spray down the wrapping and open in the biosafety cabinet. In cases in which the stock bag of dishes is opened,
whatever the glasswares we have sterilized using we have to bring it very carefully to the laminar flow. We can wrap it with aluminum foil or a cellophane bag. We can put it, right? And we have to bring it carefully to the laminar flow. Under the laminar flow, we have to open it, right? The media, after preparing the media, we have to sterilize them using autoclave, right? Once it finish, 15 minutes, we have to allow to cool, right? More than 3-4 hours. Thereafter, we have to take the basket and bring the basket to the laminar flow room, right? Then we have to open the culture vessel, right? The lid under the laminar flow, right? Each and every step here, we have to do it very, very carefully.
be sure to seal the bag inside the BSC. Correct pipetting is important for maintaining sterile conditions. First, select the pipette volume appropriate for your task. Open the wrapping and peel down 1/4 of the way. Hold down the loose flaps of the wrapper to eliminate risk of the tip touching the outside of the wrapper. Place the pipette in the automated pipetter and turn the pipette to indicate the units of measurement you would like to see. It is important not to touch the tip of the pipette to anything not sterile. Dispose of all used pipette tips in the biosafety container. When uncapping reagents, use one hand to hold the lid with the inside facing downwards. Do not leave any unused reagents uncapped. When pipetting, it may be useful to adjust the aspirating and dispensing speeds. F for fast, M for medium,
That
and
pipette
S for slow.
disperser, we can actually set the volume what we need, right? And we can use it.
When you are finished with your work in the BSC,
Once we
spray
have
down all
finished
surfaces.
that culturing work, again, we have to take out all the things what we take into the laminar flow. We have to take it out, right? And we have to again wipe the laminar flow with the alcohol, right?
With 70% ethanol. Spray down the vacuum lining with ethanol as well and straighten out prior to turning off the vacuum. Turn off the lights, blower, and close the glass panel. Thank you for watching. We hope this video clarified any questions you may have had regarding sterile tissue culture techniques.
Are you clear with the technique? Now, did you get some idea?
Yes, ma'am.
Okay. Is this visible to you, types of tissue culture, this slide?
Yes, ma'am.
Okay.
Yes.
Types of tissue culture. First one, callus culture. We have already seen what is callus, right? In the video clip also you have see that it is an unspecialized, unorganized, dividing mass of cells. Right? These are actually callus. Right? This callus actually turns brown. So it means some callus, they are different color. Some plant, it produce yellowish color callus. Some plant, whitish color. Some plant, creamy color callus. Right? And most of the callus, when it turns brown, it's the indication of dead cells. Right? This callus production and maintenance of unorganized mass of proliferative cell from isolated plant cell, that is explant, tissue or organ by growing on an artificial nutrient media in a glass vials under controlled aseptic condition. Right? What is callus? It is an unspecialized, unorganized, dividing mass of cells. When it produces actually, when we culture the explant in a growing culture media in a glass vial, right, under aseptic condition. Organ culture means, organ culture refers to the in vitro culture and maintenance of an excised organ primordial or whole or part of an organ in way and function. Right? Whatever the organ, when we used as an explant for the culturing, we called as organ culture. Right? We can use shoots, shoot tips, we can use it. Nodal, small nodal region, we can use it. Immature nodal region, mature nodal region, root tips, leaf, right? Pollen, whatever the organ we use, that is organ culture. Single cell culture, a method of growing isolated single cell aseptically on a nutrient media under controlled condition. Right? That is single cell culture. Suspension culture means actually we instead of inoculate in the semi-solid media, right? Without adding phytogel or agar, we can make the liquid medium, right? For that solidification, phytogel or agar we normally use, right? But when if we are not using agar or the phytogel, we can make it just a liquid media. It will not solidify, right? So what we can do, right? We can transfer the single cells or small aggregates of cells. We can transfer it into the liquid medium and we can keep it for the agitation. The agitator is there. We can keep it in that on the agitator. It agitates slowly. We can adjust the speed for the suspension culture, right? So from the suspension culture also, new shoots we can obtain, and also this is a suitable method for the secondary metabolites production, right? What we can do actually, we can take the callus, right, and we can just smash it to make a very small tiny aggregates, cell aggregates, and those cells, the small aggregates, we can transfer to liquid medium and we can keep it on the agitator for the further culture process, right? Embryo culture means aseptic isolation of embryo. We have to isolate the embryo from bulk of maternal tissue of mature seed or capsule, and in vitro culture under aseptic controlled physical condition in glass vials containing nutrients, semi-solid or liquid media to grow directly in the plant. Right? From the embryo, right, we can grow a direct plantlets, right? Whatever the media, semi-solid media or liquid media, we can use it, right? Aseptic condition, we have to maintain it, right? The best example for the embryo culture is maize, right? Maize embryo, that is a commonly used for the practical maize embryo. We can use it and we can observe the quick emergence of maize plantlets, right? And the culture and androgenesis, we have already seen in the importance haploid plant production. This is the one, and pollen culture, yeah, right? And the culture androgenesis, gynogenesis also there, right? Androgenesis is the ovule culture, right? That is different. Here we are going to see the culture. Androgenesis is the in vitro development of haploid plant originating from potent pollen grains through a series of cell division and differentiation, right? Here, this is the plant. We have to select the bud, right? We have to select we have to take the anther and we have to sterilize them, right? And after sterilization, we have to wash it, right? We have to cut the bud to take the anthers, right? We can cut the bud using scalpel, and we can take the anthers, right? Anthers we can directly culture on the nutrient media or culture media, right? Organogenesis is possible. If not, embryogenesis is possible. Whatever it is, we can apply the haploid plantlet. Since this is an N, right? Okay, this is anther culture. Sorry, I couldn't talk. Okay. Here, again, I'm just telling, this is a flower. We can take the flower buds, right? And we have to bring it to we have to wash it first outside, and we have to bring it to laminar flow, right? And the laminar flow, we have to do the surface sterilization. Once we finish the surface sterilization, we have to wash them with distilled water twice or thrice, right? And the sterilized buds, we have to take it and we have to cut it using scalpel. The scalpel blade is there, scalpel handle is there. All the things should be sterilized. Scalpel blade and handle also we have to sterilize using oven, right? And we have to cut it. We have to take the anthers, right? If you want, we can separate the filaments. We can cut off the filament, right? And culture only the anther into the nutrient media. From that, directly it can produce as a plant that is haploid plant. If not, it produce callus, and from the callus it produce plant that is embryogenesis. Right? This is the anther culture. What is the time now?
10 o'clock.
Oh, okay, you have lectures? Yes or no?
No, ma'am.
Shall I finish this one? These only 3 slides are there, right? I finish it.
Okay.
Pollen culture, in vitro technique by which the pollen grains are squeezed from the intact anther and then cultured on nutrient media, right? Here what we do, we can take the anther. From the anther, we can squeeze out the pollen grains, and the pollen grains can be cultured. Right? We can just look at the brief step, and they collected from the flower buds and pollen grains are isolated, and about 50 anthers are placed in small sterile beakers containing 20 milliliter of liquid basal media. Anthers are then pressed against the side of beaker with a sterile glass rod. Right? For the squeeze out, we can use the glass rod. The homogenized anthers are then filtered through nylon sieve to remove the anther tissue debris. Right? First, what we do, we can put the anther, we have to sterilize them, right? And we can put it in the liquid basal media. And by using the liquid media, it is in the beaker, right? Or in the conical flask. Beaker is easy for the squeezing. And just we can press it against the side of beaker using a glass rod. What happened? The anthers from the pollens are spaced out, right? And what we can do then, the homogenized anthers are filtered through a nylon sieve, right? Then what we are doing this step to remove the tissue, anther tissue debris. Right? The filtrate or the pollen suspension, the debris we can remove it, and the pollen suspension we can obtain it. It can be centrifuged at a low speed for 5 minutes. The supernetted contains fine debris. That one also we can remove it. Right? Only we can take the suspension. It contains the pollen, and we can wash it twice using centrifugation, right? And resuspend again in a fresh liquid media, right? And then what we can do, now the pollen suspension is ready after removing the all the tiny debris part. Now the pollen suspension is there. We can pipette out whatever the amount we need, we can pipette out and we can spread it in petri dish. Right? Most of the time, when liquid media we use, that is the best media for the pollen culture. Right? Then what we can do, we have to incubate it. Right? And from that, organogenesis or embryogenesis, whatever it is, we can produce. That one also haploid plant. Right? Next one is the protoplast culture. Protoplasts are the cells in which cell wall has been removed. Right? Those cell wall removed cell we call the naked cell. That is protoplast. Right? They possess plasma membrane and other components of cell, but those cells do not contain cell walls. Protoplasts are potentially totipotent individual at the single cell level. What did I mean? From the protoplast, we can produce a whole plant. That is the meaning of totipotency, no? So they have the potential to regenerate a cell wall, de-differentiate, divide mitotically, and form unlimited growing cell clones. Right? Differentiate shoot and root meristem or embryos which regenerate whole plant. It is the culture of isolated protoplast which is potentially capable of cell wall regeneration, cell division, and growth and plant regeneration. Right? Absence of cell wall, but that protoplasts are capable to produce the cells with the cell wall. Those cells which can be go for the cell division, growth, multiplication, and regeneration. Right? Then shoot tip and meristem culture. Tips of shoots can be cultured in vitro, producing clumps of shoots from the axillary or adventitious buds. Right? We can choose the shoot tips If not, shoot tips or the apical meristem for the shoot tip culture and meristem culture. Right? This is, we know, this meristem culture, not shoot tip culture. Possibly that not all the shoot tip culture, meristem culture, they are free from virus. Right? So stages of micropropagation, we can study in the next class, right? What about the free time? Do you have any free time tomorrow morning? Any free time tomorrow morning? No? Don't you have any free time?
Miss, tomorrow also 9 o'clock we have class agronomy.
Okay, then 9 o'clock I will take the lectures, right? Sometimes if I forgot to put the link, please use this today's link, right?
Okay.
Okay.